Hello dear candidates, welcome again to Bioclasses online. Today we are going to discuss the second law or the second rule of probability which is used in the genetic analysis, and the law is the multiplicative law or the product rule. It is also called as AND rule, and according to this product rule, the probability that two or more independent events will occur in a particular order is equal to the product of their individual probabilities. So, this product rule states that when two or more independent events occur in a particular order, then the probability of these events is equal to the product of their individual probabilities. Now let's take few examples to know how this product rule is applied in the genetic numericals or genetic problems. The first example is a normal couple's first child has a cystic fibrosis. What is the probability that their next two children will not have the disease? So the numerical is asking that there is a normal couple who had a child suffering from the cystic fibrosis disease, and you are asked that what is the probability that their next two children will not have the disease or are unaffected. As we know, this cystic fibrosis disease is inherited as a recessive trait. This means that this disease can occur only when the recessive allele is in the homozygous condition. As we already know from the numerical that a phenotypically normal couple has already produced an affected child, and to be affected, a child must be homozygous for the disease allele and thus has inherited one copy from each parent. I will repeat here again that there is a phenotypically normal couple who has already produced an affected child, and to be affected, a child must be homozygous for the recessive allele or the disease allele, and thus this affected child or this cystic fibrosis, the child suffering from the cystic fibrosis has inherited one copy from the each parent. So we can say here that the parents are heterozygous carriers for this recessive disease or for this cystic fibrosis, and with this information, that is the information that the parents are the heterozygous carriers, and we can calculate the probability that they will produce unaffected offspring. Now let the capital N is the allele, normal allele, while as the small n is the recessive allele or the disease allele. And if we will try to draw a Punnett square, the male will produce the two types of the gametes because being as heterozygous. So one of the type of the gametes will be carrying the normal allele, and one of the type of the male gametes will be carrying the recessive allele, that is the small n. In the same way, one of the type of the female gametes will be carrying the normal allele, that is the capital N, and one of the type of the female gametes will be carrying the recessive allele, that is the small n. And if we will cross these alleles with each other, in the first square we will get the capital N, and in the second we will get the capital N, small n, and in the third square we will get capital N, small n, and in the fourth square we will get small n. And you can see here from this first square we have capital N. These offsprings are normal and are possessing the homozygous and are in the homozygous condition because possessing both the alleles are the normal allele. The second offspring, that is the square second, here the offspring is genotypically heterozygous carrier, but phenotypically they are normal. Again, so this is normal phenotypically, this is normal and the third is third offspring or the the offspring which we have shown here in the Punnett square, in the third square, again they are the heterozygous carriers, but they are phenotypically normal. But the fourth square, in the fourth square, the recessive allele or the disease allele exists here in the homozygous condition, is present in the homozygous condition. As I already mentioned that this cystic fibrosis is inherited as a recessive trait. That is, this disease can occur only to those individuals which are having this recessive allele in the homozygous condition. So only out of these 4 offsprings, one only this is the offspring, this is the offspring, this one is the offspring which is affected, while as this first, second and third, they are the unaffected offspring. So this shows that this is a case of the normal monohybrid cross and showing the ratio, showing the 3:1 ratio, and by means of using, as we are using here the Punnett square, this couple should produce a ratio of 3 unaffected, 1 affected. So if we will try to calculate now the probability of a single unaffected offspring, so from here the probability of a single unaffected offspring is how many offsprings are which are unaffected from this Punnett square? 3, and the total number of the offsprings are 4. So the probability of single unaffected is the 3/4, and to obtain the probability of getting 2 unaffected offsprings in a row, as we have been asked what is the probability that their next 2 children will not have the disease or will be unaffected. So to obtain the probability of getting the 2 unaffected offsprings in a row, that is in a specified order, we must apply here the product rule. So the probability of getting the single unaffected child is 3/4, and to obtain, or we can say the probability of that their next two children will not have the disease will be 3/4 into 3/4, that will come into 9/16, or we can say 0.56, or this 0.56 can be written as 56. So from here we can conclude that there is a 56% chance that their next 2 children will be unaffected, or their next 2 children will not have the disease. So coming to the next numerical or next example, the next example is a cross has been made between the 2 plants of genotype. There is a cross has been made between the 2 plants having the genotype of capital A small a, capital A small a, capital B small b, capital C small c, capital D small d, and the second genotype is capital A small a, capital B small b, capital C small c, capital small d small d. So a cross has been made between these 2 genotypes, and you are asked that what is the probability that the offspring will be of the genotype of small a, small b, and small c, and small d. So here in this numerical we have to assume that all the genes assort independently, and if we assume that all the genes assort independently, then we can do this calculation easily by using the product rule. And the 4 different gene pairs are considered individually as if 4 separate crosses. There, as you can see here in the numerical, that there are the 4 separate there are 4 gene pairs. The 4 gene pairs are the first gene pair is this one. This one is the first gene pair. This is the first one, this capital A, this is the first gene pair, capital A, small a, and capital A, small a. Second gene pair is small b, capital B, small b, and the third gene pair is small c, capital C, small c, capital C, small c, and the fourth gene pair is capital D, small d, small d, small d. And we have these 4, we have to assume that these 4 different gene pairs individually as if 4 separate crosses, and then the appropriate probabilities are multiplied together to arrive at the answer. So let's try to consider the first gene pair, and if we will cross the first gene pair, that is the gene pair this one, this is the gene, this one, capital A, small a, capital A, small a, and here we have to draw the Punnett square to know the outcome of this cross. So if we will draw a Punnett square here, the 2 different types of the male gametes will be carrying the capital A and small a. Again the female gametes will be carrying, one of the type of a female gamete will be carrying the capital A, the second one will be carrying the small a. And if we will cross here, we will get in the first square capital A, in the second square we will get the capital A, small a, in the third square we will get the cross this A, small a to this capital A, we will get the capital A, small a, and in the fourth square we will get the small a, small a. And you can see here that it is only in, this is the genotype, this is the required genotype, and you can see here the probability of getting the genotype of small a from this cross, that is the cross between the first gene pair, is how many offsprings are having the genotype of small a from this cross? 1, and total number of the offspring is 1, 2, 3, 4, 3 plus 1, that comes equal to 1 by 4, or we can say that this from this cross, that is the cross between the first gene pair, 1/4 of the progeny or 1/4 of the offsprings will be of the genotype of small a. Now we will try to cross the second gene pair. The second gene pair is the small b, capital B, small b, and if we will cross them, we have to again draw the Punnett square and try to draw here the Punnett square. We will draw here the Punnett square. First we will erase here. So from this cross, first cross, we have got 1/4 of the progeny which are having the genotype of small a, and now we will try to draw the Punnett square for the second cross, and from the second cross we will write down here capital B, small b. One of the type of the gametes will be from this genotype, this one. There 2 types of the gametes will be formed. One of the type of the gametes will be carrying the capital B, and one of the type of the gametes will be carrying the small b. And from this genotype we will get the one of the type of the gametes will be small b, carrying the small b, and another type of the gametes will again be carrying the small b. So we will try to cross here. We will get capital B, small b, and capital B, small b, and fourth one small b. As you can see here in this Punnett square, we are getting here the 2 offsprings out of the 4 offsprings we are getting which are having the genotype of small b, and so the probability of getting the genotype of small b from this cross is, that is 2 divided by how many total genotypes are here? 4, that is the 4, or we can say 2 ones, this 2 ones are 2, 2 twos are 4, or we can say 1/2 or 1/2 that is equal to 1 by 2 is the probability, or we can say 1/2 of the progeny from this cross will be of having the genotype of small b, small b. So from the second cross, from the cross between the second gene pair, we are getting the probability of 1 by 2, or 1/2 of the progeny, or 1/2 of the offsprings will be of the genotype of small b, small b. In the same way, we have to cross the third gene pair, and the third gene pair is the capital C small c, capital C small c. You can see here, this is the third gene pair, and we will try to draw again here the Punnett square, so in order to know the outcome of this cross. So you can see here, let me erase it first. We will draw here the Punnett square. So you can see here how many types of the gametes will be here. One of the type of the gamete will be carrying the capital C, and another type of the gametes will be carrying the small c. And again here, one of the type of the gamete will be carrying the capital C, and again another type of the gamete will be carrying the small c. So when we cross, we will get capital C, capital C, capital C, small c, capital C, small c, and small c, small c. Again you can see here we are getting only one offspring. This is the offspring out of the four which is having the genotype of small c, small c. So the probability of getting the genotype of small c from this cross, that is the cross between the third gene pair, is the 1 by 4, is the 1 by 4 is the probability. Or in other words, we can say 1/4 of the progeny, or 1/4 of the offsprings will be having the genotype of small c, small c. This is the 1/4 is the probability. And in the same way, we can we have to cross the fourth gene pair, and the fourth gene pair is the capital D, small d, small d, small d. And if we will cross them, we have to again draw the Punnett square. Again here we will draw the Punnett square, and from this genotype we will get the two types of the gametes. One of the type of the gamete will be carrying the capital D, and another type of the gametes will be carrying the small d. And from this one we will get the gametes, all the gametes will be carrying the small d. So small d, small d. And if we will cross here, we will get the capital D, small d, here small d, small d, and here capital D, small d, and small d, small d. As you can see here in the from this cross, that is the cross between the fourth gene pair, we are getting the two offsprings which are having the genotype of small d out of the total 4 offspring. So the probability of getting the small d genotype from this cross, that is the cross, this cross, is the 2 by 4. 2 by 4, 2 are the offsprings having the genotype, and the total number of the offsprings are 4. So again 2 1s are 2, 2 2s are 4, it comes 1 by 2. So the probability of getting the genotype of small d from this cross is the 1 by 2, or in other words we can say 1/2 of the progeny, or 1/2 of the offsprings will be having the genotype of small d. And so therefore we can say the overall probability of a genotype small a, small b, small b, small d, small c, and small d will be, or is, that is, first, from the first cross we get the probability of 1 by 4, and from the second cross we get the probability of 1 by 2, and from the third cross we get the probability of 1 by 4, and the fourth cross we get the probability of 1 by 2. That comes equal to 1 by 64, and that is equal to 0.015625, or in other words we can say 1.5625% is the probability. Or we can conclude that the that 1.5625% of the offsprings will be of a genotype small a, small b, small b, small c, and small d, small d. Subscribe and share Bioclasses online. Thank you.